Hi everyone, welcome back to another anatomy video. In today's episode, we are going to be looking at the muscular system. So please like and subscribe so I can keep making content. And now let's get into the first topic of discussion, which is your muscle shapes. So the first muscle shape you have is parallel. This is where the tendons and muscle fibers are all going to be going in the same direction. So, an example of this would be your biceps brachii, or just your biceps in simple terms. Then the next one is unipennate. One example is your semimembranosus extensus digitorum. This is basically where you have a starting segment, and then all of the tendons and muscle fibers are branching out from one side of it. While bipennate, an example is your rectus femoris, is where you have the starting segment and then the tendons and muscle fibers are branching out from both sides of it. So over here is a nice depiction of that. You have your parallel muscle. You can see all of the lines are going in the same direction. Unipennate, it's branching off from one side. Bipennate, it looks a little bit like a tree and it's branching out from both sides. Then you have your multi-pennate muscle shape. An example of this would be your deltoid, aka your shoulder muscles. So you have a starting point and then it branches off into multiple segments. And in each segment, you kind of have that bipennate shape where they're branching off from both sides. Next is convergent. If you ever think of your chest area, so your pectoralis major and minor, or your gluteus maximus, this is the shape that a convergent muscle would have, where it has the point of origin over here at this dot, and then everything is connecting to that circle. Similarly, you have your circular muscle where you have 2 points of origin, one on each side, and then the muscle fibers and tendons follow in that circular shape. So, the muscle that is around your mouth, the orbicularis oris, and the muscle that is around your eyes, orbicularis oculis, are great examples of circular muscles. Next, parallel muscles with tendinous bands. So these two horizontal lines over here, these are your tendinous bands. And then all of the muscle fibers that are going vertically, this is your parallel muscle. So an example of where you could find this is in your abs, aka your rectus abdominis. And finally, you have your wrapping muscles. These are kind of the ones that make that C-shaped curve. An example is your supinator and pronator teres. These are the muscles that are in your forearm, which help you to supinate and pronate your hand, meaning that turning your palm upwards is supination and turning your palm downwards towards the ground is pronation. All right, now we are going to switch a little bit more into the skeletal muscle organs and the different layers that are surrounding all of the different parts that go into the muscle organ. So you can see over here that we start off with a whole skeletal muscle. So this big piece over here, that is a skeletal muscle. And around it, we have this layer called the epimysium. It's spelled right over here, epimysium. And again, epi means superficial, so it's going to be the most superficial layer. Within the skeletal muscle, you see all of these tiny little circles, and there's actually one of them is pulled out over here. So, this second projection over here, this is a muscle fascicle. And around muscle fascicles, you're going to find paramecium. So, it goes epimecium and then paramecium. And then finally, if you were to take out yet another projection, it would now be a muscle fiber. And around the muscle fiber, you would have endomysium. So, endo refers to something that is the most deep, and therefore it is the last layer. So, it goes epi, pen, and then endo. A good way to remember this is there is the mnemonic EpiPen. So EpiPen. And the epi stands for epimysin, the P stands for paramycin, and then the last EN portion stands for endomycin. So that is a good mnemonic to help you remember in which order the layers go. All right, now moving on, we are going to look at a whole muscle fiber. So this whole model is a muscle fiber, and you can see over here on this on the back of the model, it even says this model is a muscle fiber. Each one of these little circles within, so this projection over here, that is a myofibril. So again, myofibril right over here. Now, within the myofibril, you have a couple structures. I'm going to go on this zoomed in version so that it's easier to see. First, you have your myosin and actin. So, myosin is going to be these thick vertical lines that you see running along the top over here. Again, that is myosin. And then the thin lines that are in between the thicker ones, that is going to be your actin. So over here is one actin, there's another actin, and these kind of just alternate and go hand in hand. Now you have your Z lines, which is this hot pink little squiggly area over there. So that is a Z line. And if you could see a little bit deeper on this model, which you can see on this region over here, you can see that if we look at this area, you have one Z line and then another Z line down here. From here to there, that is a sarcomere. So Z line to Z line equals sarcomere. And I'm going to put this over here so it's easier to see against the white background. All right, Z line to Z line is a sarcomere. Now getting back over here, we see that we have our triads. So these three lines over here, the white, orange, and then white again, that is your triad. Within the triad, you have two terminal cisternae. I'm going to move it over here so it's easier to see. So again, two terminal cisternae. The terminal cisternae are the white lines. So this white line up at the top and then this white line at the bottom, that is your terminal cisternae. And then the last part of the triad is the T-tubule. So you're going to have one T-tubule, which is this orange line running down the middle. Plus one T-tubule. And again, the orange one is the T-tubule. I often think of it kind of looks a little bit like a hot dog to me. The white, aka the terminal cisternae, is like the bun of the hot dog, and then the orange is the hot dog itself that is in the middle of the buns. You can also kind of think of it as like a sandwich or something like that. Then you have this white webbing, that is in the center, so this white webbing over here, that is your sarcoplasmic reticulum. Right over there. Looks very much like a spider web. And sarcoplasmic starts with an S, spider starts with an S. That could be a good way to remember it as well. Moving back to this whole big model, there is a couple other accessory structures that you have to know. The first of which is this big cell over here. You can see that it's labeled as number 22. That big cell is a myosatellite cell. Right over there. Myosatellite. Now on the side, this purple structure over here, this is going to be your nucleus. So the muscle fiber does have a nucleus in order to kind of code for all of the instructions. Then this side piece over here, that is your neuromuscular junction. So this right over here, neuromuscular junction. Within the neuromuscular junction, you have your mitochondria, which are these purple little squiggly guys over there. They code for all of the ATP so that your muscle has energy in order to move and function. And then finally, these tiny, like yellow neon dots over here. So, all of these guys, that is acetylcholine. And acetylcholine is extremely important when you are looking at the sliding filament theory, which basically describes how muscles are going to contract. So, in the sliding filament theory, the acetylcholine is going to go to the binding sites within the sarcolemma, and then eventually it is going to cause these Z line to Z lines, so the sarcomere, to shorten, which is how your muscles tighten and contract. I am going to make a video on the sliding filament theory, so if you guys have any questions on it, because I know it can be very complex, then you guys should go watch that video and just comment down below if you have any remaining questions afterwards. All right, now on the outside, so this is looking on the outside of this muscle model, you need to know the two layers. So this tan layer, this whole portion over here, that is your sarcolemma. And then on the outside, where you have this kind of purple, it also looks a little bit webbed. That is going to be your endomysium. Endomysium. And that is kind of like the wrapping around everything. All right. And then a very last picture. This is just like a side picture of this model. This curved looking structure, that is what your neuromuscular junction looks like from the side. So, neuromuscular junction, aka NMG or NMJ, sorry. All right. Now we are gonna go over this lovely model over here, which I know looks very intimidating and very confusing, but it honestly is not that bad. So this whole thing is a myofibril. Now, this red squiggly little portion, that is going to be your Z line. And now we have one Z line here, another Z line here. And as I explained up above in the other model, Z line to Z line equals a sarcomere. So this whole portion over here, sarcomere. Now you have your myosin versus actin. So your myosin is going to be the dark blue. So dark blue equals myosin. And then the little like red portion, the thinner ones, this is going to be your actin right over there. Now, one thing that gets a little bit confusing is your I bands versus your A bands. So, A bands is going to be talking about your myosin. So, basically, the A band is myosin to myosin. So, this portion to this portion. And I can label that from here to here. This whole thing, an A band. Now, in retrospect to your I band is going to exclude all myosin. So your I band is going to start over here, and then it's going to keep going until you reach another myosin band. So let's say that there is another dark blue line starting here. That means that this middle segment where the actin is, that would be an I band. In this model and in this picture, you cannot see another myosin band. And I know that in a lot of anatomy classrooms, they use this same picture and they have it on a laminate. And it can get very confusing, kind of differentiating where the A versus the I band is. Basically, they just cut it off early, and that is why you cannot see a full I band. But hypothetically, if they did show another region of myosin over here, then where the next myosin would start, that would be the end of the I band, because I bands, again, exclude all myosin. So that is really important to know. Another really great way that I like to remember, whether a band is in charge of myosin or actin, is because myosin is always going to be the darker color, and the word dark has an A in it, so an A band. Now, actin is the lighter band or the lighter color, and lighter has an I in it, so I band. I feel like that is a really good way that helped me whenever I would forget about which band has which structure and which substance within it. So I hope that this kind of helped explain all of that. And now in the sliding filament theory is where conceptually this picture is really important, because these Z lines over here in red, they're basically going to come closer together. They're going to constrict. It's going to shorten this sarcomere, and that is your actual muscle contracting, which is what the sliding filament theory is talking about. So I hope this really helped. Please leave any questions, comments, concerns in the comment section. And again, please like and subscribe for more content. I also am an anatomy tutor, so if you want some extra help, then just comment and I will reach out to you either through the comment section or an email and just like be able to coordinate with you. Again, thank you so much for watching, and I will see you in the next episode. Goodbye.